Welcome to the fourth session in Cognitive Neuroscience. Today the topic will be vision and attention. First, I will give you an overview of the neural basis of visual processing on the retina level, then on the cortical level, and then we will be talking about attention. I will talk about a few paradigms such as the visual search task and also some theories and models that are relevant in visual attention research, and then eventually we will talk about neural measures of attention. The visual data stream from the eyes to higher cortical areas has many steps, but the most important steps are the retina, the LGN in the thalamus, and the primary visual cortex. On the right hand side you can see how the signal from the eyes crosses at the optic chiasm. This is where the lateralization of visual signal starts. In the left hemifield is represented in the right hemisphere and vice versa. But both areas, the right and left hemisphere, get input from the left and right eye. Let's start with the retina. So the retina is actually part of the central nervous system, and it has various cell types that are important as a first visual processing step. The weird thing about the retina is that the light receptive cells, the cones and rods, are actually not on the outer layer. So the light comes from this side and it trespasses these layers until the photosensitive cells, the rods and cones, are reached. Here you can see a microscopic photo showing the same thing in a less schematic version. So the light is coming from this side and the receptors are here. There are three types of cones that each respond most strongly to a very specific wavelength. So there's this cone type that responds most strongly to this wavelength, and these cones correspond to these wavelengths and are most activated by light in that frequency spectrum. Any color in the visible range has a distinct response pattern for the three cone types. So for example, if you have a wavelength of say 600, then you will see that the blue cones do not respond at all. The green cones respond to this much, and the red ones respond this much. So from the given activation pattern of the three cone types, the visual system can derive which wavelength is present. The missing of one cone type can cause color vision deficiency. So maybe some of you have seen these tests that test for color blindness, the Ishihara test. Of course, these are not the ideal lab conditions where you would normally test this, but if you're now at home sitting at your screen and if you see a 71 here, there is some indication that you might have a red-green deficiency. Without any color deficiency, what you would see here is a 74. Similarly, if you have the yellow-blue deficiency, you can see a 4 here. Without that deficiency, you would see a 42. The way these tests work is that the luminance of these dots here, for example, and here, these dots have the same luminance as the background, this orange-brown tone. So the only way to tell apart these dots here that belong to the 4 from the background is using the U, the wavelength information, rather than the luminance. So if you cannot do that because you miss, for example, one cone type, you would not see any difference between these dots here and the background. You would, however, see a difference between these dots here and the background because the luminance of the one is different from the background. The similar thing happens here. The 2 and the background have the same luminance, so the only way to tell apart the 2 from the background would be hue information. Apart from cones, there's also rods. Rods are not color sensitive, but they respond more strongly to light. The distribution of rods and cones is different. So, as you can see here, the rods are mostly in the periphery, whereas the cones are mostly in the fovea. So, the retina, as I said, is part of the central nervous system, and the retina is important because that's the first instance where a physical signal, the photons, are translated into the neural language. A very important concept in order to understand how the visual system works is the receptive field. A receptive field is a part of the visual field in which a stimulus can modify the firing of a neuron. So, for example, if you have an observer here and you look at a specific firing rate of a neuron, then you will see some spontaneous activity here, and this is related to this receptive field in this case. So the neuron is connected to this receptive field, and if you don't present anything here, there will be no change in the firing rate, but if you present a stimulus, then there will be an increased firing rate. Now, of course, receptive fields are a bit more complicated than that. They do not merely respond to the presence or absence of a stimulus in their area, but rather what they have, for example, is an on-center and an off-surround. What that means is if you have some kind of baseline activity, some sporadic firing rate here, then if you present light in the center, the firing rate of the neuron will increase. If you present the light in the surround but no light in the center, then you have even less activity than in the baseline. So the activity is suppressed here. And if you present light in both, you will have something in between. There's also the opposite, an off-center and on-surround receptive field. And here you would observe the opposite pattern, of course. So if you present light in the center and no light in the surround, then you would see a suppression of the baseline activity. If you present the light in the periphery and no light in the center, you would have the highest firing rate. And if you present light in both, then something in between. There are also receptive fields that are color sensitive, and they have complementary colors. So, for example, a receptive field here would particularly strongly respond to yellow in the center and blue in the surround. This one does the opposite, and these receptive fields are more sensitive to differences in green and red. A very important principle at the retinal processing level is convergence and sensitivity. So convergence describes how many photoreceptors are connected to one ganglion cell. So how many photoreceptors in principle can activate a single ganglion. And as you can see, for cone receptors, they have a much lower convergence compared to rod receptors. So for cone receptors, it's on average 6:1. In the fovea, the area of the retina where the visual acuity is highest, this ratio is actually 1:1. This is how it is depicted here. So only one cone receptor can activate a ganglion cell. For rods, it is a much higher convergence. So on average, 120 rods can activate one ganglion cell. It is depicted here for 3 rods, just to illustrate the principle. So what do we have if we have a low convergence? So for example, if one cone cone receptor corresponds to one ganglion cell. That means we have a very high spatial resolution, because if, for example, only this cone receptor here is activated, so there's input to this cone, then we can differentiate that from input to that cone, because they both have their own ganglion cell. As a result, we have relatively small receptive fields, and as another result, we also have a very low light sensitivity. That means that only if the cone receptor here receives enough input, enough physical energy, like photons, will this ganglion cell fire. It is not supported, so to say, by any neighboring cone cells. For rods, it is quite the opposite. So here we have a high convergence and as a result a low spatial resolution, because if this ganglion cell here is activated, we do not know whether the input comes from this rod, this rod, this rod, or in reality from any of the 120 rods that are connected to a ganglion cell on average. So even though it has a very low spatial resolution, it does have another advantage, namely it is more light sensitive. That is because it is more likely that this ganglion cell fires if a sufficient number of rod cells get input. So even though one single rod might not get enough input to activate the ganglion cell, there is a higher chance that a sum of the, on average, 120 will have enough energy to activate the ganglion cell. So, in sum, the higher the convergence is, for example, for these here, the lower the spatial resolution and the higher the sensitivity. If the convergence is low, we have a high spatial resolution but a low sensitivity. So how do photoreceptors respond to darkness and light? If you don't present anything to the cone, if they're in the darkness, then they actually hyperpolarize, which means that they release more glutamate. If you present light to a cone, then a cone hyperpolarizes and actually produces less glutamate. So at first this seems a little bit counterintuitive, because if you present more to the cone, it fires less, but it will become clear in a second how this is important for the visual system and computations on the retina to work. So you've seen that there are bipolar cells between the photoreceptors and the ganglion cells, and there's two main types, and they respond differently to the neurotransmitter of photoreceptors, namely glutamate. The on-bipolar cell, if you present glutamate to it, you will have a hyperpolarization. The off-bipolar cell, this one, will depolarize if you present glutamate to it. And as a result, you have, if you present glutamate to the on-bipolar cell, you have a decrease in neurotransmitter. If you present glutamate to an off-bipolar cell, you will have an increase of the neurotransmitter. So let's have a look at how this works if you present light to a photoreceptor here. So if you remember, the cone, for example, here is hyperpolarized if it's presented in the light, and then if this is an on bipolar cell here, then you will see less glutamate response results in the bipolar cell depolarization, so more transmitter is transferred to the ganglion cell here. If we have an off bipolar cell, this will hyperpolarize as a response to less glutamate, and there will be fewer transmitters, triggering less likely an action potential here at the ganglion cell. If we present nothing to this cone, then we will have a depolarization of the cone. If you remember, the cone releases more glutamate. But again, the bipolar cells respond differently. The on bipolar cell hyperpolarizes in this case, so releases fewer transmitters, whereas the off bipolar cell depolarizes because of the glutamate, and there's more transmitter released. So in this case, this ganglion cell would fire, this one would not fire, and here this one would not fire, and this one would fire. And if you remember, we have these receptive fields with the on and off, so with the on center and off surround, or vice versa. And this is realized with lateral cells. So, of course, there's not just a single cone. There's a neighboring cone here. And in this case, if this cone is presented in the light and this cone is in the darkness, then we have something called a horizontal cell. And the horizontal cell will release GABA if it's in the darkness. And that means that there's even less glutamate available now. So this kind of increases the whole effect that this ganglion cell fires more and this ganglion cell fires less, because the GABA inhibits glutamate. And here it's the opposite, so we have the cone, this cone in the center is not getting any information, it's in the darkness. This cone is in the light, so here the cone hyperpolarizes and there's less GABA, so there's no inhibition of the glutamate. There's even more glutamate now. So again, the whole effect is enhanced. This ganglion cell would have an even lower firing rate, and this ganglion cell an even higher firing rate. So by presenting one cone in the light and another cone in the neighboring cone is in the darkness, then we have the maximum firing rate here, and here we would have the maximum firing rate if this cone is presented in the darkness and the neighboring cone in the light. Now it is very important to note that already at the retinal level, the ganglion cells are not just firing the more light you present to them, the more stimulation you have, so they do already respond to very specific light patterns. For example, this ganglion cell will only fire if you have a very narrow, confined light signal. So, for example, if only one photoreceptor is activated, this is an enhancement, this is an activation signal. So this means that the ganglion cell might get some activation, might even fire, but not very likely. If there's light emitted on all three of these photoreceptors, you can still see they're activating, enhancing. So we have the maximum signal here. If there's more light, we have inhibiting connections here. So that means the signal is decreasing again. And then if we have even more light, the ganglion cell might even be suppressed. So this is important because that shows us that already at the retinal level there's an activation of the ganglion cell possible for specific light patterns, rather than the more light you have, the more likely it will fire. Throughout the visual system there is an M and P pathway, and this starts already at the receptive fields in the retina. So if we have very small receptive fields for cones mostly, then these are going through midget bipolar cells and then end up at P-type ganglion cells. P stands for parvus, which is Latin for small. So this is mostly for cones, and then they are transferred to the parvocellular layer of the LGN. We will talk about that in the next part. And this makes up around 80% of the photoreceptors in the retina. For the large receptive fields that are basically that constitute of very many different photoreceptors, cones, and also rods. Then we have these parasol bipolar cells because they look a little bit like a parasol, and then they are connected to M-type ganglion cells. M stands for Magnus, Latin for large, so this can be rods and cones and often combined. They are then connected to the magnocellular layer of the LGN. Okay, so far we have talked about visual processing on the retinal level, and in the next part we will talk about how the information is from the retina is going to the cortex and then is computed more sophisticatedly there.